Dr. Jaimie Henderson is a distinguished member in the Stanford faculty, and he's both a neurosurgeon and also a researcher. And as a researcher, he's done some really groundbreaking work. One example of this is he's created a device that really facilitates and improves the surgical procedure in helping patients with Parkinson's disease get a deep brain stimulation. And this method has really been extremely helpful in improving the surgical process. And many people in the world have adopted this procedure since Jaimie's developed this. And in another line of work, Jaimie has really helped people who have paralysis move and speak again. And this is really important work that hopefully in the future will become something that people can use and apply widely to improve people's lives. So with that, let's welcome Jaimie.
Okay, great. Thanks, JT. That was wonderful. Thank you, Tamar, for inviting me to come. Oh, up? Just better? You see me? All right. Okay, great. Well, thanks everybody for coming. This hopefully will be sort of fun and entertaining. We'll try to leave some time for questions afterwards. I'd like to sort of cover a broad waterfront today, talking about both advances in some of the more applied aspects of brain-computer interfaces, but also discuss some of the scientific insights that we've been able to learn because of the access that we've been granted to the human brain because of our projects attempting to restore function to people with paralysis and communication disorders. I have some disclosures. I'm affiliated with a few companies. Some of you may have heard of this little company in the Bay Area. So I'm a consultant for Neuralink, have stock options, and then we just co-founded this company to help push forward my work in, our work of a large group of us in DBS for traumatic brain injury, which we think has a lot of promise. So the hope is that we'll be able to fund our next clinical trial based on this. Okay, so anybody in the room know who this is? A few of the older folks maybe do. Okay, so this is Christopher Reeve. Christopher Reeve played Superman in the movies. And so he was sort of the epitome of strength and cool, and unfortunately was involved in a horseback riding accident where he fell from his horse and had an immediate C1 occiput dislocation and was left completely quadriplegic and had to be on a ventilator because of the respiratory impairment. So if we look at his brain, obviously, I think as most people would know in this room, his brain is perfectly fine. In fact, he would give very moving lectures. So all of his cognitive apparatus is intact. But the real problem, of course, lies here, where the signals from the brain can no longer make it to the limb. So a question that neuroscientists asked for a long time was, what happens to the cortex in this situation? Does it just go away? Does Wallerian degeneration just sort of make this all shrivel up? Does it reorganize? What happens? And it turns out that we now know, after more than a decade of work in people, that approaching 2 decades now, my goodness, that indeed information is still captured in this area of the brain, and the intention to move is still present. We can decode these signals, feed them through a computer, and then route them back to the outside world in order to do useful things like run a prosthetic limb, a functional electrical stimulation system, a computer cursor, et cetera, et cetera. And so this has been done successfully by a number of groups over the years. In my laboratory, we use this small sensor called a Utah array. So this is 4x4 or 3x3 millimeter array of electrodes. This one happens to have 100. We've also used the 8x8, which is a 64 electrode. And these 1.5 millimeter long electrodes penetrate just into the outer layers of the cortex. This is actually 2 scale with a cortical slice, demonstrating the scale of the electrodes to each of these cells. And each of these electrodes can pick up action potentials from one or many neurons in the brain. And so this allows us a very rich representation of a small patch of the cortex. So of course, these are very tiny signals. So these require routing out through a pedestal, which attaches to the skull. Unfortunately, we still have a transcutaneous device. We'd love to make a fully implantable one at some point in the future. A preamplifier box goes on top of that, which allows us to amplify those very tiny signals and then send them out to be processed by the computer. And this is a spike panel showing the individual action potentials from the 96 active channels in one of our research participants. So this is actually from participant T5. And what you can see is that a fairly good number of channels have these pretty large action potentials. Some of them don't record very well, which is unfortunate, but interestingly, those channels may come in and out over time, and you may get more signals from one versus another over a period of time. So if we look at this panel of spikes, and let's look at an individual channel, which has, actually a couple of different spikes on it, but let's just look at the raw threshold crossing. So this is just, we set a threshold, which is usually minus 4.5 RMS. And whenever a spike crosses that threshold, we record that as an event. And then if we look at what happens to that channel during, for example, here, this is a delayed reach task, an 8-target center out task from the lab of my dear colleague, the late Krishna Shenoy, showing that this monkey is fixating on a central target. A target appears, he moves towards that target, and then the activity in his brain is recorded during that movement. And this has been described many times over, originally by Georgopoulos in the 1980s. This phenomenon called cosine tuning, where reach directions are represented in individual channels. Each one of these are neural rasters. Each one of these little dots is a spike. These are all different trials, and this is time. And as you can see, with leftward movements, this channel fires much more rapidly. With rightward movements, it fires much less rapidly. And so this rate encoding actually makes a cosine-shaped tuning curve that each individual neuron represents. So you might think, because we're in a very small patch of cortex, which is maybe 3 millimeters or maybe 4 millimeters, that each of these preferred directions would be pretty similar across the entire array because it's a small patch of cortex. But it turns out that this is not the case. In fact, all of these different channels are tuned differently, and sometimes tuned to different types of movements or tuned to different body parts, and we'll explore that a little bit later in the talk. But if we denote the strength of the firing by the size of the arrow, we can sort of represent any movement by firing rate. So if we ask our participant, whether it be a person with paralysis or a monkey, to move a cursor up and to the left, we see that those neurons that are more highly tuned up and to the left will fire more. Those tuned to the opposite direction will fire less. And by reading out the ensemble activity of all of those neurons, we can fairly accurately decode the person or the experimental animal's intent. And so this is an example of, I don't know if the volume is high enough for you to hear this. You can hear the click in the background. So this is a 65-year-old woman with ALS, Lou Gehrig's disease. It's a degenerative neurological disease which causes her to lose function of her limbs. She still does have some retained dexterous movement of her upper extremities, and actually could move her hand fairly well, but we've asked her to suppress her active movement and just think about moving the cursor. So she's controlling this cursor by thinking about moving it around this optimized keyboard, and we're asking her to answer the question, how did you encourage your sons to practice music? She's a luthier. She made guitars and violins and violas before she succumbed to this terrible disorder. And so she's typing out her answer in a free-form way, and across the bottom you can see a sort of a real-time count of the correct characters per minute that she's able to achieve with this interface. And it's sort of hovering right here in between the 20 and 40 range, with an average of somewhere around 36 to 38 correct characters per minute. So we published this back in 2017 at eLife. This is a very nice graph by my colleague Eddie Chang, published in JAMA in 2019. He's up at UCSF, the other institution in the Bay Area other than Stanford. demonstrating the speed of various communication prostheses. And one of the things that our laboratory has been very interested in is restoring communication. So as you can see, we leverage these motor signals in order to provide assistive technologies to restore people's communication ability. And so here we are, sort of at this 2D cursor BCI, and this is right here at this BCI-driven cursor control stage. So it's 40 characters per minute, or about 8 words per minute if you count 5 letters as a word. And so as we can see, there's a whole bunch of real estate over here to the right of the curve, where we could maybe anticipate something like a QWERTY touch screen, or eye tracking is highly used by people with paralysis, or something like handwriting. So it turns out that we can actually do this. And so this is work that we published in 2021 in Nature, demonstrating that we can, in fact, move this curve more to the right by decoding handwriting. And so this is what the system looks like. So we read out from, this is our subject T5 with 2 arrays implanted in the hand area of the motor cortex. We read out those signals. We type text on the screen and then ask him to imagine handwriting. So has very little movement. He's a C4 quadriplegic, so he has very little movement of his extremities, but he's imagining, or attempting, he's actually attempting, not just imagining, and this is an interesting, important concept I'll be happy to talk more about afterwards. But he's imagining a yellow legal pad, and he's writing individual characters on this yellow legal pad. So we ask him to write A, B, C, and we have him do this over and over and over again, just writing individual letters and trying to make them in a stereotyped way, so that we know how those are represented in the brain. And so the question that we asked here was, does the neural representation of handwriting remain intact years after paralysis? And so here's the task, as I said: prepare, go, move, and then prepare again. So just keep going through these various different letters. And it turns out that, in fact, handwriting is preserved. So this multi-joint complex movement can be extracted from motor cortex. So can we reconstruct the pen tip trajectory from the neural activity? It's pretty cool that we can actually do this. So these are the instantaneous velocities that we obtain from the neural firings over time, plotted as characters. And so it's really somewhat remarkable how much these characters resemble the ones that he's trying to write. So this is sort of a one-to-one representation of handwriting in the cortex. So that's cool, but what can we do with that? So this is the system that the folks in our lab, particularly Frank Willett, who is the lead author on this study, absolutely brilliant postdoc in the lab, now research scientist. So this is those threshold crossing features that I talked about, just looking at thresholds of each individual channel. This is the entire electrode array of 192 channels, 192 channels over time. And so we move a sliding window across these threshold crossing features, and we take each of these time slices that represents an individual letter, and we train a multi-layer GRU recurrent neural network to learn the relationships between those threshold crossing features and the individual characters that our participant intends to type. And so what that outputs is a series of probabilities that the RNN says, over time that it thinks the probability of any individual character being typed. And then a second output, which is transition probability. So the probability that there's a transition between a new character. If anyone's interested, I have an entire illustration of this pipeline. I thought I would leave it out for the sake of time here, but if anybody's interested in the details of this, I can talk more about it later. Okay, so then we take these outputs, and then we generate a raw output from this, which here you can see there are some errors. So for example, here it's, instead of the paper, it's TNE paper, because N and H, of course, look very similar. And are often confused. And so the second part of this system is a language model. So this is a trigram language model that's optimized via Viterbi search offline, and then applied prospectively into this retrospective output from the language model. So this can be run in real time, as the interface is being used. And so we assess this in a closed loop fashion with our participant, and these are the results. So the video that I showed you previously was about 40 correct characters per minute, and that's this dotted line down here. And over 5 days of use, T5 was able to improve his performance. And this is mainly just Frank like saying, go faster, go faster, go faster, from about 65 characters per minute up to 90, essentially 90 characters per minute. This was actually a rather uncomfortably fast speed for him. He would prefer to write a little bit slower, but he did this because we egged him on to do it. And he's a very competitive guy. He said, I'm going to beat your monkeys. Anything your monkeys can do, I can do better. And he's serious about it. And then this just shows the error rates. So interestingly, as I demonstrated in the previous slide, even though the neural network's pretty good at doing this, it still has a lot of errors. And this can change day over day. This is one of the sort of flaws or problems with intracortical interfaces, that there's day-to-day instabilities. And that's one of the things our lab is really interested in helping to pursue and figure out. So this is the raw output in circles, and you can see it varies anywhere from about single-digit errors, below 5%, all the way up to double digits, maybe 12. But with the language model, and this is just, again, a very simple trigram language model. This is not one of the new, GPTs. We can get these error rates down close to 0. So that's pretty cool. And it's actually very usable. So on the bottom, yes, sure.
A bit about how the decoder is framed. This is a quadriplegic.
Right. Right. So he's attempting movements. So we'll have, I flew past the task design a little bit quickly. So there's a letter appears on the screen. There's a red box, meaning prepare to write the letter A, for example. Green box comes on. He writes the letter A. Then the red box comes on, says prepare to write the letter A. He prepares. Green box comes on. He writes the letter A. And he does this over and over and over again, hundreds of times. It's pretty tedious and boring. But in the end, it provides enough data to train the RNN decoder. But there are many more steps. I mean, it's not as simple as that. Well, anyways, I could spend a long time going this. I won't. But suffice to say, there's a hidden Markov model. There's time warping and stretching. There's synthetic data that we augment because it's, RNNs are very data hungry, and this is a data poor regime. So there's a bunch of steps that we go through in order to improve the performance of the RNN. So I think that might have been what you were getting at. You say, how do you train the RNN? There's a lot of stuff that goes into it, but I'm going to gloss over that in the interest of time because I want to get through, hopefully we can get through quite a bit of stuff. But, if not, we'll keep it on time. We'll stop if we have to. Okay, so on the bottom is that same sort of video that you just saw with our participant with ALS, and this is T5, our participant with spinal cord injury, who's using that same interface to just do copy typing. So just say, imagine all the people living life in peace. This is nice because it provides a good metric that we can use to compare different decoders or different paradigms. And so this is a similar performance to what you saw before, which is actually, when this was developed, it was a step function better than what there was before in terms of cursor control and typing speed. And then, so just as a comparison, I'll run that same video again on the bottom, but I'll run a handwriting video on top. And so because the RNN can't really detect an absence of something, so we have him kind of artificially make a bright caret in order to indicate the space between words. And so you'll just see the two running head to head. And so, as you can see, he's already ahead doing it with the handwriting interface. It's not perfect. And this is part of the downside of having an open loop, a relatively open loop paradigm, is that it's hard to correct errors in real time, which you can do here with the delete and space keys. But this is, as you can just see visually, is about double the communication rate of what we were able to show with the cursor-based communication processes. Okay, and so again, this is over 5 days of use. accuracies, again, this is the full pipeline with the language model incorporated, really hovering around between 91 and 96%. And then just to reiterate, these characters per minute up around 90, which is now about 15 words per minute in comparison to the 8. So this is essentially double what we were showing with the cursor-based control. And of course, he can use this similarly to how we showed you T6, using it with answering open questions with the interface. He can do the same thing with the handwriting interface. As you can tell, he doesn't eat very healthy. I ask him, what's his favorite meal for dinner? So he likes mostly fried food. He loves old movies. So, there's a network in the US, TCM, Turner Classic Movies, which is nothing but old movies, Bogart films and such. And, he loves to watch this on television. So he can answer questions with this as well. So I at one point had a slide of Stephen Hawking, just, the Lucasian professor, a lot of tradition there. I didn't include that here. It just didn't quite fit. But he was, of course, famous for using his very low bandwidth communication prosthesis, which really worked on cheek twitches. But I sort of think to myself, what if we could provide a prosthesis like this, for somebody who's lost the ability to communicate in an effective way? And how many of you have ever read this book or heard of it? Just a few. I recommend this actually it's a really cool and interesting book. It was written by Jean-Dominique Balbi, who was the editor of Elle magazine, suffered a brainstem stroke and was left with the ability to only move one eye. And he wrote this entire book, character by character, by having an assistant scan across a letterboard and moving his eye when she reached the correct letter. So this entire book was written character by character in that painstaking way. Again, it's a short read, as you might imagine, but it's unbelievable to understand the quality of life that people have who you wouldn't really expect. You would think, well, this is a terrible, devastating thing, but he actually really loved life, and so being able to provide the ability to communicate for people like that, I think, is a huge goal. Okay. So let's move on then from upper extremity control to something that I think is even more interesting and cool, which is the neural basis of speech. I don't know. I could watch this forever. It's amazing. It's unbelievable what we do with our articulatory muscles. All of the actions, the complicated things that we do that just go completely unnoticed. We take it all completely for granted, but this is an incredible motor control problem. And, fun to study, obviously, because of this. But of course, number two, which really should be number one, is this translational need for speech BMIs for patients who can't speak. Okay, so here's our Intrepid T5. He's the one who's going to beat our monkeys. So we had another weird task for him to do. We said, well, okay, we're going to have you do something really weird, Dennis. And that is, we're going to have you talk, and we're going to see if the hand area of your cortex represents phonemes, words, orofacial movements. He said, sure, I'm game, whatever you want to do. And so this is what we had him do. So we would, again, here's the trial design. We would prompt him to say a syllable or a word. We used a very small subset of words and syllables and orofacial movements. We'd say, for example, say ba, then there would be a go click, and then he would say ba. And again, so this is a representation of where his electrode arrays are. So here's the precentral gyrus. This is a more dorsal portion of the precentral gyrus. This is an area of the cortex described by Usry as it's called the knob, which is a very reproducible area that represents the hand. Okay, so if we look at an individual neuron of this array or individual channel, and we can sort spikes. So this is a sorted spike. This is an actual neuron, not just a threshold crossing. And we look at how that neuron responds to these various stimuli. This is a PSTHs that just show what those spikes or what the firing rate looks like over time. Here's the go cue when we ask him to speak. Here's voice onset time is right here. And you can see that there's, I was pretty surprised to see how much tuning there is for speech in the dorsal part of the motor cortex in an area far away from anything that we would expect speech to be in. And again, if we look at the individual phonemes or syllables that are represented here, it's not subtle. You don't need statistics to see that there's modulation at voice onset. And this particular neuron really seems to like k and ah and certain other various phonemes, and really doesn't care too much about oo or e. If we just choose another channel, let's choose this guy from over here. This one really likes k. Again, similarly, there's tuning to individual phonemes or syllables. And then if we look across the various different types of articulator movements, including movements of the mouth and lips, movements of the tongue, we can see that these neurons are tuned differentially to these various different movements. So again, these are those same two neurons. And this guy, really likes tongue up movements and tongue down movements. It doesn't really care too much about, tongue left or right. But neuron 2, of course, likes these things very much. And so if we look across the array to the responsive cells, the ones that modulate their firing rate, there are a number of electrodes that respond only to syllables, not necessarily to orofacial movements, which is, again, interesting and kind of weird. But many neurons that responded to both types of stimuli. So again, this really was consistent with some of the prior work that's been done in ECOG that suggests that the motor cortex is important for controlling speech articulators, but this is a little bit outside the area where these other investigators were looking. So that we found that very interesting. And then this is just to show that using a multi-class support vector machine, this is a fairly simple, straightforward classifier. We can classify these different classes. It's a small number, so it's not a hard classification problem, but the fact that we can do it at all is actually pretty cool. Chance here is about 10%. And so we're doing way better than chance, and actually something that could be decodable. Not good enough for speech prosthesis, but proof of principle that allowed us to then sort of de-risk this idea of decoding speech from the motor cortex and allowed us to move on to the next phase of what we were planning to study. So this is something I'll return to in a little bit. This was, again, Frank Willett and then Daryl Deo, who's a postdoc in the lab, published in Cell in 2020, that in fact, in the arm-hand area of the motor cortex, there are multiple representations. So there's not just arm and hand, but there's also mouth, face, tongue. As one might expect, the arm is more strongly represented, but there's also head movement, leg movement, eye movement, tongue movement, and even higher dimensional control. So, so 4D cursor control actually had its own unique representation in this area of the brain. So, clearly the motor cortex, in this case, premotor, this is not M1, we're in the crown of the precentral gyrus, and we'll talk about this in a little bit, where this is actually really premotor cortex, I think, can do some really very interesting things. Okay, so what's going on here? I mean, is this some sort of plasticity or is it remapping? I don't think so, because if that were the case, why would the arm and hand still be the most strongly represented things here? You would expect for it to be remapped almost everything to the head and face, which is not the case at all, actually. So probably not really what's going on. I think it's closer to this, that the single neuron somatotopy is more distributed, more overlapping than what we see with either microstimulation, which is where the original studies were done by Penfield et al., or electrocorticography. So this is a more complicated picture at a neural ensemble level than what we see at a higher level of recording. So this, and again, as I said, this is proof of principle for a speech neural prosthesis. And so this is our implant plan for participant T12, who I'll show a little bit. So 2 arrays in the ventral precentral gyrus and 2 arrays. So this is area 6V. So this is premotor cortex, and this is in area 44, which is what's sometimes called Broca's area. Although I'm not going to talk a lot about it, but there's something interesting about Broca's area in this participant. Area 44 does not care about speech, which is kind of surprising again. Okay, so we based our implant plan on the terrific work done by, I mentioned Eddie Chang earlier. He's our collaborator, friendly competitor to the north at UCSF. We have a great relationship, so I can say that. But of course, his work is really groundbreaking and really set the stage for much of what we do. And so This is a paper that he published in Nature. Bouchard is the first author here, looking at somatotopy in ECOG in people with epilepsy who are admitted to the epilepsy monitoring unit for epilepsy, and showing the somatotopic representation of articulators throughout the precentral gyrus. So, for example, GA is represented more ventrally, as you can see by the red electrodes, and BA is represented more dorsally up here. And so we decided that we were going to base our implant on this, hedging a little bit in terms of trying to find somatotopic representations of articulators, but thinking that we probably weren't going to need to, care that much, just given what we saw in the dorsal motor cortex. And so this is our plan for our implants. If there's is anybody bothered by pictures of surgery or blood or anything like that? Okay, this is warning, okay? Don't look away, because there will be a surgical picture that appears now. I'll tell you when it's gone, okay? So anybody online, you can just turn away or whatever. So this just shows an intraoperative photo. This is a video of me localizing these. So I performed the implant surgery on all of our patients. So this is me localizing the array implant locations. This is the IFG, or inferior frontal gyrus, Broca's area. So this is the superior and inferior arrays. And then this is ventral precentral gyrus. And this corresponded really very nicely with our preoperative plan. As you can see here, there's a blood vessel that's in the way of this array. We didn't want to hit that. So we had to move this guy a little bit north compared to where we had hoped to move them. And so this is where this is the final picture of where those implants ended up. Okay, and then so if we have a look at, okay, Tamar, you can look. So now if we if we now look at the arrays, I'm actually pretty proud of this. This is almost every channel is recording here. So this is actually we gave a big cheer when we saw this, because we wait a month before we begin recording and we never quite know what we're going to get. So this was really pretty cool. So again, just looking at individual channels. So let's pick a channel at random. This guy, channel 47. You can see there's a couple of neurons here, but if we just look at threshold crossing rates and the fractional variance accounted for by that individual channel, the darker colors represent more variance, or higher levels of tuning. And so if we look at, for example, forehead movement, wrinkle your forehead, move your eyebrows, this has pretty broad tuning across that superior array, but really pretty disappointing in the more ventral or the more lateral one. So yeah, if we're basing our ability to decode on this, of course, we have to look at other things like the other articulators, jaw, lips, tongue, larynx. And again, way better performance at identifying these different articulators in the more dorsal part of the array. But something really interesting turned up, and that is that the more ventral array represents more complicated things. So phonemes and words are more strongly represented in the more ventral part of the precentral gyrus, as opposed to the more dorsal part. So there's this differentiation, again, sort of violating this Penfield homunculus model, in which there's this speech hotspot in the more ventral part of the precentral gyrus. So, again, this is sort of a foreshadowing. We're going to talk a little bit more about this if we have time. Okay, so again, this is the pipeline that I just showed you for handwriting. We repurposed it almost identically for speech. It required a little bit of tweaking. There are some things that we had to do for speech that we didn't have to do for handwriting that were a little bit interesting, because we're decoding phonemes instead of characters, and so they're represented a little bit differently, but it's functionally the same thing. So here's our threshold crossings, and we also use spike band power because it helps with the decoding. And we throw those into the RNN. So again, similar multi-layer GRU. And then these are the phoneme probabilities that we extract. Not quite as clean, as you see, as what we got with our letter probabilities with the handwriting prosthesis. Again, language model, same trigram language model. By the way, I wish I forgot to include, there's a QR code that I generated for this. We have a competition, actually. We've released all the data for this online. And so we're challenging people around the world to do better than us at decoding, and hopefully people will come up with better decoding strategies. So if anybody's interested, I can share that with you, the website for that competition. And then this, again, the same thing. It's just essentially a sophisticated autocorrect. Okay. And so here's the system in use. So this is our participant T12. She is a 67-year-old woman with ALS. It's actually bulbar onset ALS, meaning that she's, it started in her brainstem and then moved to her extremities. So she still retains some ability to do some movements. She can still type on an iPad, and she usually uses a stylus. She can handwrite on a tablet. She can still get around. She uses a scooter, so she's still living independently, but she's very dysarthric. So as you'll be able to tell from the video, she can't really generate intelligible speech. She sort of makes noises, but it's not intelligible. So she uses a lot of prosody, a lot of tone of voice in order to communicate. And of course does a lot of typing, but that's a very painful, slow, and laborious process. So here we're having her do just essentially a copy task, just like we had our other participants do with handwriting. And she's just supposed to read this, and then the output from the decoder in real time will happen along the bottom. And then once the entire sentence is done, she pushes a button. She's still able to do that to say, I'm done with the sentence. And then the text-to-speech decoder or text-to-speech system then just speaks it.
I don't want to call her a babysitter. That would be good. I did well in school.
So once the system gets rolling, it works pretty well. Of course, there are errors.
I don't see much pollution turning on.
And in full disclosure, I sort of cherry picked, a nice run. So they don't all look like this. This is a particularly good
That's
run.
it a lot of the time.
Interesting. It seems as if after a few trials, things at the very beginning, things are pretty rough, and then after a few trials, this tends to clean up. Okay. Of course, we can also use the system to ask her free response questions, and then have her, what we had her do here was to then type out what it was she was, or what she was trying to say. She didn't like to do this very much because it required sort of a pause in the session for her to indicate what it was she was saying. But you can sort of get the gist here. So works pretty well. This is also interesting. She doesn't necessarily have to phonate, so she can just mouth the words, and the performance is actually the same, or maybe even a little bit better, because it's not as effortful for her. It takes a lot of effort for her to produce sound, and so by just using her speech articulators without the diaphragm and larynx, actually tends to improve things a little bit.
I do not have much to compare it to.
So yeah, it works in that regime as well. One of the grad students from the lab is actually taking this a little bit further and looking at
I don't read as much
inner
as I would like
speech
to
or
either.
imagined speech, as well as overt speech. And it turns out that we can decode that as well, although at a much lesser accuracy than overt speech. So there's even representations of inner speech in this region of the brain. Okay, and so here's the results. So, I showed you, we started at sort of around 8 words per minute with the cursor control. We ended up at about 15 words per minute with handwriting, and then arrive at about 65 words per minute here. And so the word error rate's still unacceptably high for daily use. This is, like I said, I showed you a very nice segment of this, but it hovers right around 24% or so, 24, 25%, which is 1 word in 4. Usually you can guess what she's trying to say, but yeah, I wouldn't call this not quite yet usable. Again, I have another video from our collaborators in the BrainGate Consortium at UC Davis, who have taken some of the things we learned from this and done the next thing, and it is very impressive to see. So this was the first proof of principle demonstration, but it's nowhere close to the theoretical limit of the performance that can be achieved. And then this was really interesting. This was a fully general English vocabulary, so 130,000 words. Most people use maybe 40 to 60,000 words if they have a very large vocabulary. This includes it's something called the switchboard corpus. It's made up of telephone conversations that have been transcribed. So it contains, a lot of words that are, morphemically similar. It's just they have different morphemes, but the same root. Different suffixes or prefixes, but same root. But regardless, this is again at about 65 words per minute. And then if we drop this down to a much lower vocabulary that somebody might use if they were just wanting to do communication for things that they needed on a rapid basis, we can drop that word error rate down to single digits and maintain a pretty high words per minute encoding. Okay, so again, just to show that we're continuing to increase both speed and accuracy over time, and the hope is that someday we'll be able to get up here. But interestingly enough, that was her comfortable speed of communication. She didn't want to try to talk any faster than that, and in fact, she probably couldn't because she had adapted over time to this communication style. So perhaps if we could intervene earlier in the course of the ALS and get people using something like this earlier on, we might be able to get closer out to this, even faster communication. Okay, so in the last 10 minutes or so, I don't know, we probably want to leave 5 minutes. Okay, I'll run through this pretty quickly. So, this is, I showed you this bar plot about motor representations in the arm-hand cortex that really span the entire body. And the idea that single neuron somatotopy is more distributed and that this, classical representation is actually somewhat incomplete. And, if we look at how things are organized or what things are encoded in the dorsal precentral gyrus, it's actually interestingly different from what we see in the ventral precentral gyrus. So, instead of this, variability across different limbs, et cetera, with some differentiation between left and the right. Here we see that things look pretty similar, except for face and speech in the ventral precentral gyrus. So Daryl Deo, who was the co-author on the cell paper that I showed you before, postdoc in the lab, has taken this on as his pet project, and I think it's really cool what he's come up with. So it turns out that there's probably an organization that looks something more like this, where there are really 3 subsegments to the precentral gyrus. There's a middle precentral gyrus, which is area 55B in the Human Connectome project, and that Eddie Chang has shown. If you resect this area, it acts kind of like Broca's area. If you get this expressive aphasia, and so this is probably very necessary for speech. The dorsal precentral gyrus, which is involved in bilateral limb control, and the ventral precentral gyrus, which represents things in a bilateral way because it's mainly involved in speech and in sequence encoding. Okay, so we've been able to do this really cool thing because we're part of a larger consortium called BrainGate. We can actually enroll multiple participants with intracortical electrodes, this is the first study that's ever demonstrated this number of participants. So we have 1, 2, 3, 4, 5, and then we're able to convince the University of Chicago to collaborate with us. So we have a couple of arrays from their participants as well. So this is a total of, here's the number of arrays, where they're placed in each participant, and just to show that we've got a really nice span that we can look at throughout precentral gyrus. So in order to assess this, we use the same task across all participants, which we call it the whole body sweep. It's a very simple suite of tasks that we ask people to do in order to just briefly assess the tuning to the various different body parts. And so this is just kind of an example. You know, we have them speak a few words, we have them move some articulators, we have them turn their head, left and right ankle, and these are all implanted in the dominant motor region. And so just as an example, so we'll pick an example channel. This is from participant T5, our monkey beater, who it demonstrates, as you might expect, that right hand is represented in this dorsal precentral gyrus most strongly. In the middle part of the precentral gyrus, speech. This is in participant T15 at UC Davis. And then T12, which is one of our participants, in the superior part of the precentral gyrus, tongue movement or articulator movement, and in the ventral part, more speech again, as we talked about. So, really interesting and cool differences. And so this is a complicated and confusing thing here. So I'm going to try to break it down so that you understand what's going on. So this is that's that whole body sweep. And so these are each of the individual movements that are included in that whole body sweep. This is the modulation strength normalized across all the arrays. How much does any individual neuron modulate, with 1 being the most and 0 being the least. And what you can see just by briefly scanning this chart visually, and each of these represents one array and one participant. So this is all the different arrays that we have implanted across the span of the precentral gyrus. And what you can immediately appreciate is the ventral part of the precentral gyrus here is very heavily speech. And as you get more dorsal in the precentral gyrus, that speech drops off. But then you begin to get a broader sort of tuning in T12 or in T15. We believe that this is actually, these more superior ones are representing a different sort of class of information. In the middle part of the precentral gyrus, T15, again, very strong speech encoding and a little bit less of other things. T16, who has another one which we had hoped it hit area 55, probably turns out that this is in prefrontal eye fields, and this is the clue right here, head movement. We didn't actually assess eye movement, so this is probably an eye field. And then, interestingly enough, as you get to the dorsal regions, you get this bilateral representation. So right arm most strongly, but also right leg, left arm, and some left leg as well. These X's are channels without significant modulation. Okay, and then this just kind of shows that same information in a different way, but it's compressed. Each of these is an individual participant. Each of these is an individual array, just showing the span from the inferior frontal sulcus, superior frontal sulcus are indicated here. So this is the ventral, mid, and dorsal precentral gyrus. And again, you see this sort of pattern where this speech-related array here in the middle of the mid part of the precentral gyrus matches what we see more ventrally, and more dorsally. We see this with this representation of bilateral things. And I'll skip this slide, but this is just to show that. And I can explain this more if people are interested, but if you look deeper in the sulcus, you get more specificity, and on the crown of the gyrus, you get less specificity, again, corresponding probably to the area 4 here and to area 6 here. Okay, so I'll wrap up, leave us maybe a couple of minutes for questions. Sorry, hopefully I didn't go over too long. I hope I've convinced you that brain-computer interfaces are moving closer to clinical reality. I personally believe that they are. We're almost there, with companies like Neuralink, Paradromics, others building better and better interfaces with some of the fundamental neuroscientific findings. I think we're within 5 years of having devices that could be deployed in people's homes to help them restore function. And to me, that's really exciting because this has been something we've been working on for a long time. Because we have this opportunity, these arrays that we're using to help people, this also enables pretty cool fundamental human neuroscience that we can do. So the future is very exciting. I think this is just a great time. These are the people who did the work. I just get to stand up here and brag about everything that they did. Here's Frank Willett, amazing guy we talked about. Daryl Deo is right here. Again, terrific work from him. Here's some of our grad students that have done a lot of the speech speech-related work. Nishal Shah, he's hiding back here, doing a lot of the upper extremity work, and then collaborator Shaul Druckmann, and then our computer people and clinical neurotechnology research assistants. And then this is Aaron, who's also doing a lot of the speech work. And again, we're part of a larger consortium, which enables a lot of this work across many different sites. And this is, everybody as of about 2 years ago in the BrainGate Consortium. So thanks. And with that, I'll try to take some questions. Okay, so yeah, I think we'll try to alternate, I guess, between Zoom questions and online or offline questions.
Any questions?
The quick question, even within modality for example, for handwriting, so not just imagining the own handwriting, but someone else's handwriting or even just letters, would it work? And the second one, is there any relationship between the severity of physical, physical difficulty and the success of the decoding?
Second question first, no. It doesn't seem to relate. If somebody can be completely paralyzed and still have excellent decoding, that's T5. Some people with retained movement may have worse decoding, and it really doesn't seem to correlate. We actually train our decoders on the first question, which was, can just by watching, do you see modulation here? And we actually train our decoders. It is attempted movement. So we have them watch a task being performed in open loop and have them try to mirror that task. But much of this information can be derived from just observation. So there's actually a couple papers from others in the group within the consortium looking at the differences between watch, imagine, and move or attempt. And it appears that those are really almost just sort of scaled versions of one another. So there's representation of watching as well as moving. Yeah.
Zoom questions.
Yeah, Zoom questions. Let's see. Nothing yet. Okay.
Well, I guess I'll ask a somewhat related question to the last one. So you talked about in the speech case, there's a copy task, right? And there's another internal representation task where the participant kind of answers a question and does she type out the actual thing afterwards?
Afterwards, she lets us know if it's correct or not.
Okay. I was just interested in, is there any difference between the performance of a decoder in the two kind of training paradigms?
Very similar.
Okay.
Presented gyrus is separate passes rather than as some sort of gradient, or indeed some other region of interest, because the very first decoders showed an array of electrons populations with sort of an overlapping code for direction based on relative activity. And I wonder what led your decision to treat the precental gyrus as three separate regions, each coding independently, as opposed to the larger instance of this overlapping.
Yeah, so all of that stuff that I showed you about that is all hot off the presses data, by the way. Paper's not even been submitted yet. So that's really preliminary stuff. We came to that. And I tried, hopefully I sort of conveyed how we got there. We started with the dorsal precentral gyrus, which seems to encode upper extremity as we would expect. We found speech decoding there. So we were like, well, now we should go ventrally because we want to decode speech. And that was our goal with our participant with, anarthria, is to say, well, we don't care so much about upper extremity. We want to be able to restore her ability to speak. And then as we looked at the ventral precentral gyrus, this discovery that there are two separate regions there, that there's a more speech articular related dorsal region and a more speech related, just simply speech, phonemes and words more ventrally, got us interested in this whole idea that there's this discontinuity. And it turns out that that's actually the case. And this is bolstered by research by a really nice paper that I didn't get a chance to show. That was the last slide I skipped, from Matt Glasser and David Van Essen's group at WashU, using Human Connectome Project data to look at the various different modes of where these regions lie along the precentral gyrus. And area 55B, which is this area in the middle, appears to be different from either the ventral or the dorsal. And so we just started discovering over time, as we investigated this more and more, that there appear to be these different functions throughout the different regions. And so that's what led us to investigate this. And in fact, the findings from our participant T12 then informed T15's implant, T16's implant, and is also informing T17's implant. So we're using the information that we learned to refine our implantation strategy for participants to come. Does that answer the question?
Definitely explains sort of how the problem should be solved in months, deciding where to put an implant. That's your ultimate goal, is to sort of find a small area of cortex representing the most detailed information. And I was wondering whether, I guess phrased in a different way, whether we can then conclude, all right, yeah, it's definitely the right place to put an implant. Can we infer from that 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 is where the information is represented, or could that just be the sort of binding hub region of a more distributed information representation? That is, so of course you've got a hedge of bets by placing in the apparent most of the region,
Sure.
but actually it's not a holistic.
No, I think I'm sorry. Oh, wow. Okay.
Whether decoding means representation.
Yeah. Does decoding mean representation? No, I think that I think that in fact, that is what's happening. I think that these are hubs of a more distributed representation, but obviously there are concentrations of these things. There are still representations, like, for example, of phonemes and words outside of this really concentrated region, but this is where that appears to happen most, I guess. And so, yeah, but to me, the interesting thing is that there are these differences, that it's not just a strict sematotopic mapping from lateral to medial, which is kind of the dogma that we've been taught over many years.
Maybe one last question.
I'm curious about the activity of first, how much retraining do you need to do with impressions, and you already have a lot of data?
Yes. So this is also being written up, hopefully will be published for much longer. We've looked at all of the arrays across all of the brain gate experience and 16 participants or 15 participants. And it turns out the performance is quite variable over time. Some of them roll off quite rapidly. So the question for the online people, sorry, is do you see array performance changes over time? Is there day-to-day variability? Is there roll off in the array performance? All of these things do happen. So there is instability. Single unit recordings are unstable. And there could be a number of reasons for this. It could be electrode motion. It could be gliosis. It could be, we think that things may move from day to day. Neurons may come in and out. We may lose a neuron one day, pick it back up again another day. So we have to compensate for this. And that's one of the research areas that our lab's been quite interested in how do you recalibrate in an automatic way from day to day. If you don't automatically recalibrate, you have to do that in a manual way, which is time consuming and sort of a pain. But that is one of the downsides of the intracortical approach as compared to something like ECOG. It is more unstable, but we also think that the performance is worth it. It's a little bit like a modern fighter jet, where they're fly-by-wire because it would be impossible to fly it without the computer. And that's because they're aerodynamically unstable, but that also increases their performance. So in kind of a similar way, this is how I think of intracortical recordings, is they're more unstable, require a computer to help you, but the performance that you can eke out of it is better.
So it's a little past 3 p.m. People who need to go can of course go, but people want to stick around.
Yeah, I'm happy to stay for as long as people have questions or.
Yeah. It seemed like there were a few other hands up.
So I have some supplemental slides if anybody cares.
So why don't we give a hand?
Yeah, let's give a hand. All right, we're going to the second half.
Okay.
No.
Oh, yeah, nice. It's my pleasure. Appreciate the invitation.
Next time you come back, you'll be on the right side.
Okay.
The scale. You'll be off the charts to the chalkboard.
Yeah, it'll be nothing but questions.
Online questions?
Yeah.
We'll take some online questions.
Oh, okay.
Yeah.
Great. Yeah, for some reason, I've lost the ability to see this. I don't know why. Oh, I see why. Stop share. This is why.
I don't know if this.
Oh, there are no. There are none, at least in the chat. Oh, there's a Q&A. So how should I answer this? Just talk over here. Okay.
I have a few people first.
Okay. So yeah, I guess a couple online questions. One is, was there a difference between speech decoding from thresholded versus spike sorted neural data? So we didn't actually do decoding with spike sorted neural data. And the reason being is that there are a couple of papers from Krishna Shenoy's lab. It was funny because, Krishna always believed that you had to sort spikes in order to get the optimum performance. And so a few of his grad students sneaked around behind his back and did a comparison study showing that threshold crossings were every bit as good as sorted spikes and much, much easier to use. And so we use threshold crossings because of that, simplicity and the fact that they represent information just as well. We also use spike band power. So this is high frequency LFP in the sort of greater than 200 hertz range. So that also helps with our decoding. So yeah, so that answers that, hopefully. And then a question from iPhone. Mentioned that since sharing your data openly, some groups have achieved even higher decoding performance. Can you say more about what they did to achieve this and how that has impacted your approach? It's mainly been using better language models, as one might expect. So, even with our own interlab research, in fact, another group took the record and we took it back. So I think we're on the leaderboard as #1 still, but I haven't checked in a while. And so this is just, better and better, using GPT-3, using GPT-4. We've dropped that 25%, 24% error rate down close to single digits. There's a video that I didn't show, but this is actually in review, and will hopefully, it's actually on arXiv, on bioRxiv or medRxiv. And the authors are, the lead authors are Stavisky, Brandman, and I think the lead author is Nick Card. And this is in their participant T15, who has all 4 of their arrays in ventral precentral, well, 3 arrays in ventral precentral gyrus and 1 in area 55B. And his performance, one of the things that we suggested, we did a channel dropping experiment in our original paper, speech decoding paper, in which we projected a curve. Look, the reviewers made us take out the projected curve, but we projected that if you increased the number of channels, it was a log linear relationship, that the more channels you got, the closer to 0 you got with your error rate. And so they now have 4 very nicely recording arrays, and they have achieved single digit error rates on a full vocabulary over time. One of the problems is that our participants' arrays have rolled off fairly severely over the past, she's now a couple years into the study. And so we can no longer support decoding at that high level. But their participant is able to actually use this with single digit decoding accuracy, decoding errors to communicate. And it's his now preferred method of communication. He uses it to talk to his family. He uses it to talk to loved ones and people online. He uses it to do his work. And, the video is actually really impressive. So, maybe I'll show it at some point, but you can go online on medRxiv and you can see it online. It's really cool. So yeah, I think we got off time. We were talking about threshold crossings and we ended up there. So, yeah.
Just to introduce that.
Hi.
Wondered if you could give us any more of a sense of like, what are the codes? So you mentioned there's a whole bunch of processing and some Markov modeling and so on. So I was
Yeah.
really interested, like, are we talking about like instantaneous codes, like at some particular point in time, you can tell me what code it is? Or is this really about integrating through
It's-
time?
Yeah,
And
it's,
like, if so, what's kind of time scales?
yeah, so we use 20 millisecond bins and we go, five, we iterate, we update the decoder 5 times per second. So and then there's an integration step at 1 second intervals, basically. And so that's what you saw as the letters were, so once per second, we're looking at the phonemes and then coming up with the entire word. Yeah, there is an integration step that's involved. It's a multi-layer GRU.
I think that has implications for how we should be doing our computer science sequencing. In a typical class, we're decoding B, whatever thing we're interested in, and typically we're using instantaneous or near enough, like small integration windows, but nothing up to a second because we usually try to say something about the time.
Sure. Well, I mean, we can say a lot about it. I mean, if we want to say something about the neural activity over those smaller time scales, 20 millisecond bins are pretty, about as small as we usually care about. So we can look at that nearly instantaneous, if we look at PSTHs or something, we can see peristimulus time histograms. People don't know what that is, but in order to look at the instantaneous firing rate over time to do neuroscience, but in terms of decoding, we just want as much information integrated over time as we possibly can in order to make the decision on what to decode. Yep.
Fabulous. Thank you very much.
Thank you.
We do, like many other things, decoding of semantic representations out of the bench intuitive. And actually, we rely on it evolving over time. And in fact, we do the same decoding out of our computational models. So whether it's like Jaimie psychology tomorrow. I guess my question was about what some of the plots you have, which are lovely, sort of, moving beyond Penfield plots. There's a sense in those plots where one is looking for a rather one-to-one relationship still between a sensor and feature. And presumably in the decoding models, they're not doing that at all. They're saying, just give me the data, I'm going to treat that as a distributed representation, and I'll pick out features that are predictive.
Yeah.
So I'm sort of wondering whether there's almost like a ghost of Penfield still in some of those plots of like, maybe it's completely distributed. And I thought it's asking some of my PhD students, I thought she was going to ask a standard question, which is, maybe it's not what we call axis aligned. So actually these features are distributed, have a distributed representation.
Yeah. So the question for those online is, is this, are these features quite specific to regions, or is the representation more distributed? I fully believe it's the latter. I mean, I think there's everything is everywhere in a way. I showed you those modulation depths for individual, individual arrays and individual participants, but you can pick out channels within those arrays that may respond to something. It's just that, looking as you get to a coarser level, that's sort of more, congruent with what we would expect to find, in a homuncular representation, then I think you get to that. But sort of within array or between arrays, I think we're seeing very distributed representations. And so, we're looking forward to, as we get more and more arrays placed in people, we're now we have permission to do 6. So you can then really begin to look at these inter-area interactions and, causality.
Whether that might have practical neurosurgical ramification, which is ultimately you might not need such high density arrays. If it's truly distributed, there must be some sweet spot with you want coverage, but you don't necessarily need high local density of sensors.
This is actually a really interesting question and an area of active investigation as groups that are doing ECOG for prosthetics work are getting smaller and smaller. You know, the pitch is getting closer together and the electrodes are getting smaller. And, you know, is there a fundamental limit below which, you know, the averaging is just too much and you don't need to go any smaller? But from a prosthetic standpoint, it's certainly possible that a micro ECOG may do everybody as well as what we're doing with microelectrode recording. But it's a completely open question. I don't know. I think right now the performance advantage goes to microelectrodes, but who knows? You know, it may be that, and yes, from a neurosurgical standpoint, it's way easier to place a grid than it is to put these things. Very technically, it's a very fine technical thing to put these in and have them perform well. So, yeah, it's a great question, and it's one that, you know, I mentioned Eddie Chang on several occasions. He's big in the ECOG thing, and so he and I have actually had you know, sessions where we Eddie, last session we did together, Eddie asked the audience, you know, where would you place your bets, ECOG or intracortical? And, you know, neurosurgeons all said, oh, ECOG for sure.
Yeah, very close top.
Yeah.
Sticking in.
Yeah, that's right.
So get
Yep.
floating.
Yeah, yeah.
Well, I think that's a great place to end. Well, let's.
Okay, yes. Thank you. Thanks for the invitation.